Now let us see what happened with the pyruvate which is produced by the glycolysis. So we have already seen that pyruvate or pyruvic acid is first converted into acetyl coenzyme, and then this acetyl coenzyme is entered in mitochondria for the Krebs cycle or citric acid cycle or tricarboxylic acid cycle. Then in mitochondria, acetyl coenzyme is react with the oxaloacetic acid and produce citric acid, and this reaction is catalyzed by citrate synthase. Then the isomerization of citrate to produce isocitrate, and then decarboxylation of two successive steps leading to the formation of alpha ketogluteric acid and then succinyl coenzyme. The oxidation of succinyl coenzyme into oxaloacetic acid, and then this oxaloacetic acid again react with the new acetyl coenzyme and produce citric acid, and this is how it completes the cycle known as tricarboxylic acid cycle. During this cycle, the NADPH and ATP and GTP are produced, that and this NADH plus it will enter into the oxidative phosphorylation for the formation of ATP into the and enter into the electron transport system that will we see in later. During conversion of succinyl coenzyme to succinic acid, there is synthesis of 1 GTP molecules. Means when succinyl coenzyme is converted into succinic acid, there is synthesis of 1 GTP molecules. Means it produces 1 GTP molecules when succinyl coenzyme is entered in succinic acid. In coupled of reaction, GTP converted to GDP with the simultaneous synthesis of ATP from ADP. During Krebs cycle, there is production of two molecules of CO2, 3 NADH, 1 FADH2 and 1 GTP. The overall reaction of tricarboxylic acid cycle is pyruvic acid plus 4 NADH plus FADH plus 2H2 plus ADP. It's give 3 CO2 plus 4 NADH plus FADH2 plus ATP, and this reaction take place in mitochondrial matrix. During whole process of oxidation of glucose molecule, glucose product produce CO2 plus 10 NADH2, 2 FADH2, 2 GTP and 2 ATP ultimately. Now let us see electron transport system. I have told you that the NADH that produced from the citric acid cycle will ultimately enter into the electron transport system, which have certain electron carriers, and electron move from one carrier to the another carrier, and this electron transport system is present on the inner mitochondrial membrane. So let us see what is electron transport system. The electron transport system is a metabolic pathway through which the electrons are transferred from one carrier to the another carrier. It is known as electron transport system, and it is present in the inner mitochondrial membrane. You can see in this figure that it have three regions. The main center blue color region is known as electron. It shows the electron transport system, which is present inside the inner mitochondrial matrix or membrane. The left side region is shown the intermembrane space, and right side the inner mitochondrial membrane, the matrix is shown. And we know that in matrix Krebs cycle is take place, and from Krebs cycle the NADH is produced. Then this NADH entered into the inner mitochondrial matrix in the electron transport system. So this electron transport system has comprised certain following complex, or we can say that 5 complex. First complex is NADH dehydrogenase, second complex is succinate dehydrogenase, third complex is cytochrome BC1, and fourth complex is cytochrome AA3, and last complex 5, it is made up of ATP synthase. So these are the complexes which present in the electron transport system. Now in electron transport system, one molecules of NADH2 gives rise to 3 ATP, means one molecule of NADH2 produce 3 ATP, and one molecule of FADH2 gives rise to 2 ATP, means NADH2 will produce 3 ATP in electron transport system, and FADH2 will produce 2 ATP. The oxygen play a vital role in removing electron that produce and production of H2O. The phosphorylation present of oxygen is known as oxidative phosphorylation. We know that we have also discussed this phosphorylation in photosynthesis. That in photosynthesis, the phosphorylation means formation of ATP. The phosphorylation in photosynthesis take place independent on light, so it is known as photophosphorylation. Here it is independent of light, so and it take place in the presence of oxygen, so it is known as oxidative phosphorylation. Now let us see the structure of ATP synthase. The energy released and utilized synthesis ATP with the help of ATP synthase, that is found in complex 5, means the ATP which is produced by the ATP synthase, which is found on complex 5, which is known as complex 5. The complex consists of two major components, F1 and F0. The F1 is a headpiece in the peripheral membrane protein complex and contain the site for the synthesis of ATP from ADP. The F0 is integral membrane protein complex that form a channel through which proton cross the inner membrane. Here in figure you can see that the ATP synthase is made up of two components. First one is F0, which is found integrated in the inner mitochondrial membrane, and the inner side of the membrane, inner mitochondrial membrane, the F1 particle is there, which is responsible for the formation of ATP from ADP, and the F0 component is responsible for the transport of hydrogen molecule or proton inside the inner membrane, inner part of the inner mitochondrial membrane. The passage of proton through the channel is coupled to the catalytic side of the F1 component for the production of ATP. For each ATP produced, two protons process through the F0 from the intermembrane space to the matrix down the electrochemical proton gradient. We have also know that for the formation of ATP, the proton gradient is must, and proton gradient is very necessary, and we already discussed in photosynthesis that when proton gradient is breakdown, during that time ATP synthesis. So for the formation of ATP, the proton gradient is very necessary. Now let us see next topic. Next point is respiratory balance sheet. This calculation can be made only on the certain assumption that there is a sequential orderly pathway functioning with one substrate forming the next, and with the glycolysis, TCA cycle, and electron transport system pathway following one after another. Means the calculation of how much ATP are produced during this all the respiration we can calculate on the certain assumption that yes, this kind of processes like glycolysis, Krebs cycle, and electron transport system are take place in sequential manner and certain particular order, that first glycolysis take place, then Krebs cycle, then electron transport system. The NADH synthesized in glycolysis is transferred into the mitochondria and undergoes oxidative phosphorylation. None of intermediate in the pathways are utilized to synthesize any other compound, and only glucose is being respired. No other alternative substrate are entering in the pathway at any intermediate stage. Means this point want to say that the only glucose molecules are converted or respired in the respiration. So this can be asked in exam that which molecules are respired. So glucose molecules are respired in respiration. So it is necessary for all the sugars and all the molecules to first converted into glucose. For the and only glucose is entered in the glycolysis, then glucose produce pyruvic acid, and then pyruvic acid converted into acetyl coenzyme, and we know that this acetyl coenzyme entered in the mitochondria, and in mitochondria it produces NADH and ATPs. Then this NADH goes into the electron transport system where it produces ATP. So this is the overall reaction of respiration. Now net gain of 36 ATP molecules during aerobic respiration of one molecule of glucose. So this is important for the MCQ, that from the respiration of one glucose molecules how many ATPs are produced. So the answer is the net gain of 36 ATP. Means the respiration of one complete aerobic respiration of glucose molecule will produce 36 ATP. Now next point is fermentation and aerobic respiration. The fermentation account for only partial breakdown of glucose, where in aerobic respiration it is a completely degrade to CO2 and H2O. In fermentation there is net gain of only 2 molecules of ATP. For each molecule of glucose degrade to pyruvic acid, where many more molecules of ATP are generated under the aerobic condition. Means in fermentation only 2 ATP molecules are produced from the one glucose, and in aerobic respiration it produces at least 36 molecules of ATP. So you can compare between this condition. First let us see in anaerobic or fermentation. In fermentation glucose produces only 2 ATP, and in aerobic respiration it produces 36 ATP. The NADH is oxidized to NAD plus rather slowly in fermentation. However, the reaction is very vigorous in case of aerobic respiration. Now, Amperbolic pattern. What is Amperbolic pattern? The respiration and Krebs cycle is known as Amperbolic pattern. So, if someone asks you that what is Amperbolic pathway and give the examples of Amperbolic pattern. So the empibolic pathway is pathway which in which catabolism and anabolism take place. Means catabolic reaction and anabolic reaction are take place in the Krebs cycle. So it is known as empibolic pathway. Again, the important question can be asked that why Krebs cycle is known as empibolic pathway. Because in Krebs cycle the catabolic reaction and anabolic reaction both take place, and we know that what is catabolic and what is anabolic reaction. Catabolic reaction means the breakdown of complex molecule to the simple molecule and it produce ATP or energy, where in anabolic reaction it is a formation of complex molecule from the simple molecules. So, so it is known as empibolic pathway. Empibolic pathway. Now let us see the how different molecules are respired. For example, fat, carbohydrate, and proteins. We have seen already that how carbohydrate or glucose are respired. First, it go into the glycolysis, and then it produce pyruvic acid, and pyruvic acid converted into acetyl coenzyme, and acetyl coenzyme enter into the mitochondria and go into the Krebs cycle. So let us see how these different molecules are respired in different manners. Now let us see how carbohydrates are degraded or break down during respiration. So we know that in glycolysis, glucose converted into pyruvic acid, and then pyruvic acid converted into acetyl coenzyme, and then acetyl coenzyme entered into the mitochondria, where in a Krebs cycle it produces water molecules, carbon dioxide, and ATP. So this is about a breakdown of carbohydrate during respiration. Now let us see how protein and fats are breakdown during respiration. So let us see first fats. How fats are breakdown. So fats are breakdown into fatty acids and glycerol, and then fatty acid goes to converted into the acetyl coenzyme, and then this we know that then this acetyl coenzyme enter into the mitochondria for the Krebs cycle. Likewise, glycerol converted into the dihydroxyacetone phosphate, and then it convert into the glyceraldehyde 3 phosphate. Later we it form pyruvic acid and then acetyl coenzyme. So this is about fats. And now let us see how proteins are degraded or break down during respiration. So first protein is break down into amino acid by the enzyme called protease enzyme, and then by deamination these amino acids are converted into the pyruvic acid, and later pyruvic acid converted into acetyl coenzyme, where acetyl coenzyme is entered into the mitochondria for the formation of ATP, and it produces ATP, water, and carbon dioxide molecules. So this is about empibolic pathway and respiration of different molecules like fats, carbohydrate, and protein. So we discussed about empibolic pathway, which means the the empibolic pathway means the respiratory pathway is an empibolic pathway which both the reaction are take place. First one is a breakdown of complex molecules and building up or synthesis of the complex molecules. So it is known as empibolic pathway, or we can say that empibolic pathway are pathway in which both the anabolic and catabolic reaction take place. And the examples of empibolic pathway is respiratory pathway. We can say the Krebs cycle is the most important example of empibolic pathway. Now the last topic of our unit is respiratory quotient. The ratio of volume of CO2 evolved to the volume of O2 consumed in the respiration is called respiratory quotient. or respiratory ratio. So respiratory ratio is volume of CO2 evolved upon volume of O2 consumed. This can we can the respiratory ratio we can calculate by this equation. So the equation is volume of CO2 evolved upon volume of CO2 consumed. The ratio is depend on respiratory substance. So the carbohydrates are completely oxidized and the respiratory ratio is 1. The CO2 and O2 equal amount evolve and consume. For fats, the respiratory ratio is less than 1 and it is a 0.7. For in a respiration of fats, it requires 102 carbon dioxide molecules upon 145 O2 consumed for the evolution of 102 carbon dioxide, and the ratio for the protein is 0.9. So what is the importance of this respiratory quotient? So the importance of this ratio is to we can identify that which kind of substrate are respirate during this process, during respiration. So by this ratio we can calculate that, or we can calculate that which kind of macromolecules are respired in the body. So this is about respiration in plants. Now in which we discuss about respiration, what is respiration, and then we discuss about glycolysis, Krebs cycle, TCA cycle, electron transport system, and then we discuss about the Amperbolic pathway. And what is Amperbolic pathway? And last we discussed about respiratory quotient. So here we completed our chapter respiration. So thank you.